Assalamu alaikum. Hi everyone. I am Younas from University of Veterinary and Animal Sciences, Lahore. Topic of my presentation is use of immunoinformatics approach to design multi-epitope-based vaccine against Riphicephalus microplastics to control bovine babesiosis. What is ticks? Ticks are the obligatory ectoparasites. Parasites are the organisms that depend upon the host for their food, reproduction, and it transmits the various diseases to the host. Ticks cause the tick-borne diseases such as babesiosis, thalidosis, and anaplasmosis in animals, and it causes Lyme disease in humans. And it is a global threat to domestic animals and the human. Among the tick species, Riphicephalus microplus is also known as cattle tick, is the most significant in livestock industry because it transmits the babesiosis, which is a blood-borne disease and cause anemia and blood loss in the animals. About 80% of the world cattle population are at the risk of babesiosis, and annual losses due to this disease is estimated as 13.9 to 18.7 billion US dollar. How to control these ticks and tick-borne diseases? There are several approaches used to control the tick, such as chemical herbicides, biological control, and vaccines. Mostly used chemicals to control this disease, but it has several drawbacks, such as its residue passed in meat and it contaminates the environments and also develop resistance against in these chemicals in ticks. So we go for vaccines. Vaccine is a safe and sustainable method used to control these tick and tick-borne diseases. For Riphicephalus microplus in 1986, first time glycoprotein extracted from the gut of Riphicephalus microplus to control these tick. But this vaccine has several limitations and it is only effective to certain stages of ticks in some geographical areas of world. And it does not provide sterile immunity to ticks around the globe. So we go for multi-epitope-based vaccine by using various immunoinformatics tools. Objective of my research is to retrieve the sequence of potential protein candidates of Riphicephalus microplus, then prediction of helper T cells, cytotoxic T cell, and B cell epitopes of candidates of vaccines against Riphicephalus microplus, then construction of multi-epitope DNA fragments and cloning of DNA fragment in an expression vector, then expression of multi-epitope protein. Here is the methodology. First, we take the vaccine candidate sequence from NCBI database, then use several tools to predict the epitopes, then construction of synthetic DNA and check the immunogenicity, allergenicity, and physicochemical properties of this construct. After that, structure evaluation, modeling, and validation, then vaccine receptor docking, then molecular dynamics simulation analysis, and in silico cloning. How we predict the antigenicity? For the prediction of antigenicity, first we take the sequence of BM86, BM95, and Sibolicin, which are already known vaccine candidates of Riphicephalus microplus in faster format, then check the antigenicity by using scratch protein predictor, and on the basis of high scores, we select the proteins as a vaccine candidates. Here is the results. After the antigenicity, then we predict the helper T cells, cytotoxic T cells, and B cell epitopes. For prediction of helper T cells, we use Immunoinformatics database analysis to predict the HTL epitopes and selection on the basis of low percentile rank, because low percentile rank epitopes considers having high binding affinity to T lymphocyte receptor. Then prediction of CTL epitopes, we use net CTL 1.2 servers and high stored epitopes considered good binders. Then for the prediction of B cell epitopes, we use ABC PRED server, which is reported in several literatures and have high efficacy and high stored B cell epitopes used for the construction of vaccine design. Here is the result of HTL epitopes and low percentile rank of BM86, BM95, and Sibolacin epitopes used for vaccine construct design. Similarly, ABC PRED used for B cell epitopes, and on the basis of threshold values, those epitopes having threshold value of more than 0.5 will considered as a potential epitopes. And high threshold value means higher probability of inducing immunity in the animals. Then prediction of CTL epitopes. These predicted epitopes were considered more immunogenic and used in the vaccine design. Then multi-epitope-based vaccine was designed by combining 6 HTL epitopes, 8 B cells, and 8 CTL epitopes, and linked with using linker, because linker have ability to facilitate the folding of amino acid residues. And adjuvant was also added to the N-terminal, because adjuvant act as a immunogenic components and enhance the immunity in the body. After that, in silico cloning of this construct in PET28 alpha, because it is a good expression vector and reported in several experiments. Here is a schematic diagram of multi-epitope-based vaccine. First, we retrieve the sequence, then construction of multi-epitope-based design, then find the tertiary structure of that construct and physiological properties, immunogenicity, and allergenicity find by using several software such as X, which is commonly used. Then molecular docking with TLR2 receptors, and then in silico cloning. Conclusion. Our study found that multi-epitope-based protein was expressed in our laboratory. Then use SDS page and Western blotting for the confirmation of multi-epitope-based protein. And this recombinant multi-epitope protein is yet to be tested in animal model for vaccine design. At the end, I would like to acknowledge for the sponsorship to Academy of Excellence in Biosciences. And I am also very thankful to my mentor, Professor Dr. Kamran Ashraf, Dr. Muhammad Imran Rashid, and Dr. Harun Akbar. Thank you, all of you.